Hello, everyone, and welcome to today's webinar, Combining RNAscope Technology with Protein Detection, a multi-omics approach using RNA-protein integrated co-detection workflow. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Leica Biosystems. To learn more, visit leicabiosystems.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. With that, I'd like to welcome our speaker, Blake McAlpin, Field Application Scientist at Advanced Cell Diagnostics, a brand of Bio-Techne. Blake, you may now begin your presentation.
Hi, my name is Blake McAlpin. I'm a Field Application Scientist with Advanced Cell Diagnostics, the company that brings you RNAscope Technology. I want to thank Leica for having me here for this webinar. And today I'm going to talk to you about combining RNAscope technology with protein detection using the Leica bond. So the title of my talk is Combining RNAscope Technology with Protein Detection, a multi-omics approach using RNA-protein integrated co-detection workflow. In this webinar, I'll start with an overview of the RNAscope-ish technologies that we've developed at ACD. Then we'll take a closer look at our integrated co-detection workflow for RNA and protein detection using RNAscope technologies and with antibodies. I'll discuss the applications of this technique as well as the workflow. We'll also look at how to analyze results from our assays and then some common challenges and how to troubleshoot them. Finally, I'll close by answering some of your questions. So there will be a live Q&A at the end of this webinar, and I'm more than happy to answer the questions that you place in the chat while I'm giving this presentation. The key to this workflow is the order of operations. As I mentioned, some epitopes can be sensitive to the RNAscope-ish procedures. I'll get into this a little more in detail, but in this workflow, we are adding the primary antibody before the RNAscope-ish. We also include a post-primary fixation to protect that bond. After completing our RNAscope pretreatments, hybridization, and detection, we come back and add the secondary antibody and detect our protein target. This approach enables a wide variety of antibodies to be compatible with our RNAscope assays. Some of you might be familiar with the sequential co-detection protocol we've used in the past. In addition to the enhanced antibody compatibility, we've confirmed that ICW supports mostly equivalent or better performance of antibodies that were compatible with the sequential procedure. And finally, we've also tested that the ICW procedure maintains equivalent-ish sensitivity. And in this next table, I've summarized our assays that are compatible with the ICW procedure that we're going to talk about today. On the left are the manual assays, which I'm not really going to talk about today, but they're here. And on the right are the compatible ICW assays automated on the Bond RX. So this includes our LS-RED singleplex chromogenic assay, the multiplex fluorescent assay, our BaseScope assay, which targets smaller RNA transcripts, and the microRNAscope assay, which targets the smallest RNA transcripts, 17 to 50 nucleotides in length. With co-detection, we use our TSA Vivid dyes. So these are dyes produced by Advanced Cell Diagnostics, which are extremely bright and are compatible with both protein and RNAscope. Here we have a comparison of the sequential workflow that I mentioned earlier and this new integrated co-detection workflow that we're going to discuss today. You can see the timing of the protease digestion is a huge difference between the two workflows. The addition of the post-primary fixation step protects the primary antibody, and the co-detection blocker step prevents cross-detection of the secondary antibody by the ISH chemistry. Here we'll look at the impact of the ICW procedure on antibody performance. We can see good signal from the CD20 antibody on a head and neck cancer tissue sample using a green chromogen. This is the IHC only control, so there's no RNAscope on this slide. Next, we can see what happens to the signal for this particular antibody if it is applied after the RNAscope pretreatment steps. So this would be sequential RNAscope with ISH. And we can see that a lot of the antibody has been degraded, probably due to the protease. However, when we use integrated co-detection workflow, we preserve the signal from CD20, and we also have successful PPIB, these little red dots, RNAscope signal in red. Now I'm going to talk to you about a few more examples in which ICW was used and just show you kind of what it looks like. So here are a few examples of ISH IHC staining in breast cancer, lung, and tonsil tissue. So each of these use the RNAscope red assay for PPIB, which is essentially our housekeeping positive control gene. And we can see the pairing of the staining with CD3, CD8, or Ki67 IHC staining. CD3 and CD8 are T-cell markers. Ki67 is associated with proliferation and tumor cell metastasis. You can see that the ICW procedure preserves antibody signal more effectively while also introducing less background compared to the sequential staining. I'll just give you a minute to look at these. These are a few more examples of co-detection with IHC and RNAscope in cervical cancer, and lung cancer with Ki67. So we can see that there's a compatibility with both green and brown chromogens for the IHC staining. Take a look at the ICW with these red PPIB dots and the protein and compare it to the sequential staining on the left. These are some images of fluorescent ICW. So these are lung cancer tissues stained with the fluorescent V2 assay and CD3 or CD8 protein targets. You can see after the sequential staining, the protein target was pretty degraded, whereas when completing it using our ICW workflow, the protein target seemed more preserved. Here's an example of ICW from a publication from the Journal of Immunotherapy of Cancer published in 2021. This group is trying to understand the cellular source of cytokines that led to T-cell entry into the tumor microenvironment, because this can be a rate-limiting step in the effector phase of the anti-tumor response. Now, CXCL10 is very difficult to identify in tissues using antibodies, but they were able to use our multiplex fluorescent V2 kit to identify CXCL10 transcripts. Then they used ICW to identify CD45 and SOX10 positive cells with antibodies in the same tissues. They got very nice staining and were able to quantify what percentage of each of these cell types were producing CXCL10 transcripts in patients with different responses to immunotherapy treatment. It was a very interesting and clinically significant study. My last example comes from a paper published recently in the Journal of Cell Biology. This team of researchers was investigating how the loss of TDP43, which is an important protein involved in ALS, frontotemporal dementia, and other diseases, leads to disruption of cholesterol metabolism and affects myelination. Here they used our ICW kit with the multiplex fluorescent V2 assay so they could label oligodendrocytes with GSTP1 immunofluorescence and assess the related reduction of SREB, F2, and LDLR mRNA after TDP43 had been deleted. They started with the RNAscope ISH-only staining, but the data was complicated to interpret because of the other cells expressing the markers they had interested in. Therefore, they had to incorporate the GSTP1 immunofluorescence to be able to analyze the expression specifically in the TDP43 deleted oligodendrocytes, and ICW helped them to accomplish that. Now let's dig a little deeper into the actual workflow next and what you need to incorporate this assay into your research. Here you can see what reagents are required to use this workflow for the LICO autostaining platform. So there is somewhat of different reagents necessary for the manual ICW platform, but today I'm just going to focus on the LICO automated system. So included in this ICW kit is a co-detection antibody diluent specifically developed for the ICW workflow. As you can see, many common antibody diluents and blocking reagents contain ingredients that can negatively impact RNAscope. We see a lot of loss of PPIB signal in this middle image because of the composition of the antibody diluent sold by LICO. It's important to dilute all antibodies in this diluent for the ICW procedures. That includes your primary and your secondary antibodies. The typical blocking steps included before primary incubation of antibody in traditional IHC or IF protocols should also be avoided when using ICW. Here is a table that summarizes exactly what you need using the LICO assay on the right and the manual, which I'm not going to talk about much, on the left. The only important thing to note here is that the LICO assays, they all still use the ER2 as a target retrieval, and they also use the LICO co-detection blocker. So really all you need to run this, aside from some other nuances regarding the protease that I'll talk about in a bit, is the co-detection antibody diluent. Of course, in addition to the ICW reagent kit, you're also going to need a Bond RX to run this automated assay. You'll also need your chosen detection kit. So this could be RNAscope, BaseScope, microRNAscope. You'll also need your probes, including your target probe, your RNA target, your positive and negative control probes. It's important to note that if you're running more than one plexing, so 2 to 3 RNAs, your C2 and C3 probes, which are specific to your channel 2 RNA and your channel 3 RNA, will actually be 50x, and they'll be diluted directly into your C1 probe. If you don't want to use a C1 probe, you can also dilute them directly into probe diluent. Finally, you will need ACD control slides, which are used to ensure that the assay is running correctly on your machine. Other items that are required from LICO are these bond open containers. So this is what we transfer our ACD reagents into in order to run on the machine, including all of the amplification steps. You'll need bond wash, bond dewax, epitope retrieval for target retrieval, and of course your mixing station, your bond detection kits, and cover tiles to run the assay. Finally, after the assay is run, you're going to need reagents in order to dehydrate, depending on the exact detection system you're using. So please refer to your manual. For example, with fast red chromogen, we're not going to use any ethanol because it's very sensitive to ethanol and it would ruin the signal. Finally, the antibodies and secondary antibodies that you use are not sold by ACD. So these are antibodies that are used in other traditional IHC or immunofluorescent situations that are compatible also with ICW. I also just want to focus on the TSA vivid dyes. So these are fluorophores that are required for the multiplex fluorescent detection. These are sold by ACD and they are compatible with ICW. While preparing samples, it is important to note that each sample type has a varying sample preparation, storage, and sectioning guidelines. So RNAscope and ICW is compatible with both formalin-fixed paraffin-embedded tissues, fixed frozen tissues, and fresh frozen tissues. For FFPE, ACD recommends samples be fixed in 10% NBF for 16 to 32 hours at room temp. Samples are recommended to be stored at 4C for long-term storage with desiccants and be sectioned from 4 to 6 microns. For fixed frozen, samples are recommended to be fixed for 24 hours at 4C with either 4% PFA or 10% formalin, and for fresh frozen, samples are recommended to be frozen immediately after harvest. Both fixed and fresh frozen samples should be stored at -80°C for up to 3 months in an airtight container. Fixed frozen samples are recommended to be sectioned at 7 to 15 microns at 10 to 20 microns for fresh frozen samples. So this is an overview of the first portion of the workflow for the chromogenic assays in FFPE paraffin samples. Please keep in mind that the fresh frozen and fixed frozen samples will need to be treated with slightly different conditions, but both are compatible with the ICW kit. I also want to note that it is important to optimize both the ISH and IHC procedures separately for any sample type before combining them in this way. This minimizes additional downstream troubleshooting, but it is common that some optimization will be needed when combining the assays. We begin with the sample preparation I mentioned on the previous slide, followed by hydrogen peroxide pretreatment and the co-detection target retrieval. After target retrieval, we add the primary antibody, then complete the post-primary fixation. Now we can move on to the appropriate protease for our tissue and move on to hybridise our RNAscope, BaseScope, or micro-RNAscope probe. After the amplification steps have been completed and the ISH signal is developed with the fast red chromogen, the peroxide block step is performed to prevent cross-detection between the two chemistries. The protein signal is then developed using DAB or green chromogen after incubation with an HRP conjugated secondary antibody. As a reminder, both the primary and the secondary antibody should be diluted in the co-detection antibody diluent. For RNAscope, you have 6 amplification steps to build the amplification trees, and then alkaline phosphatase and fast red chromogen will be used for signal detection of the C1 or channel 1 target. You can also see an example of this staining on the right with CD19 in brown and PPIB in red. PPIB is the ISH, so this is your RNA target, and CD19 is the protein. Now the BaseScope assay singleplex is similar, but instead of using 6 amplification steps, it uses 8 amplification steps to build the amplification tree. The reason is pretty logical. It's because you're targeting a smaller RNA transcript, so you need more amplification in order to see signal. Now between the BaseScope and the RNAscope kits, the chemistry is slightly different. The amps have different temperatures and durations, so it's important that you follow the steps accurately for whichever assay you're using. The BaseScope singleplex assay also uses ALP fast red chemistry for detection of a C1 or channel 1 target, but BaseScope and RNAscope probes are not interchangeable. One is much smaller, BaseScope, and one is much longer. So if you tried to combine the two, they will not work. Again, the miRNAscope, microRNAscope, singleplex red chromogenic assay will follow a similar procedure to the RNAscope singleplex red assay, but there's an additional fixation step that must be incorporated when preparing your samples. Additionally, microRNAscope probes have a unique design that is different than RNAscope and BaseScope. So these probes are designated by an S1 in their name. And a microRNAscope probe must be used with a microRNAscope detection kit. So here you can see MIR2233P, which is in red, and you can see CD3 stained in green, which is your protein stain, and this is a cervical cancer tissue. Next, let's look at the amplification and detection for our fluorescent assays. Here, AMP1, AMP2, and AMP3 build the amplification trees for multiple channels at the same time. This is followed by sequential detection of each target. Here we have the detection of 3 different channels. This is just a visualization of what it looks like. So the amplification trees will hybridize on each channel at the same time. In addition, the amplification branches will hybridize onto these preamplification trees all at the same time. And then we go through and we sequentially deposit a fluorophore onto each channel individually. This requires the binding of HRPC1 to the C1 amplification tree, and then the addition of a vivid dye. The vivid dyes will react with the HRPC reagents to become active and deposit a fluorophore. HRP blocker is then used to block HRPC1 and end the detection process for this channel. Next, we repeat the series for the C2 tree and the C3 tree. Either vivid dyes or opal fluorophores are required to be purchased for this procedure, and vivid dyes can be purchased directly from Advanced Cell Diagnostics. This is nice because the assay has some flexibility, as any vivid dye can be assigned to any channel or any antibody. The vivid dye concentration can also be optimized, allowing for the ability to control the intensity of the staining. There are additional vivid dyes available that fluoresce at different wavelengths as well, which I will show later. They can all be used with this workflow in any channel. Next, we have the workflow for ISH immunofluorescence co-detection. The concept is very similar to that of the chromogenic ICW I just showed you, in which an antibody is added after the pretreatment steps and fixed using 10% NBF. Then the RNAscope multiplex fluorescent portion of the protocol is completed. Finally, we need to label the antibody with a secondary fluorescent. And now there are 2 ways that you can do this. Option 1 is to use an HRP conjugated secondary antibody and a TSA-based fluorophore reagent, similar to the vivid dyes that we've been using throughout the rest of the assay. And option 2 employs a fluorophore conjugated secondary antibody to visualize immunofluorescence signal. Either workflow can be used, and the selection will depend on your preferred secondary antibody. Here's an example image of the staining that can be achieved with multiplex fluorescent V2 and the ICW kit. So here's showing Ki67, which is the protein in this stain, with our positive control probes for the RNA expression, POLR2A, PPIB, and UBC. And you can see that the result is this very stunning, beautiful image that shows single cell RNA transcription along with protein marker Ki67. Here are a few general tips for success with the ICW kit. As I mentioned, we recommend establishing ISH and immunohistochemistry or immunofluorescence protocols for your samples and antibodies separately before attempting to combine them. We also know that many researchers include a blocking step before adding their antibodies, and if you would like to do this before either your primary or secondary antibodies, we recommend using the co-detection antibody diluent in place of your standard blocking reagents. Our diluent includes components that can help prevent non-specific binding. And I didn't really talk about this because we're not talking manual in this presentation, but if you are doing the manual assay, we recommend using the overnight primary antibody incubation as the breakpoint of the assay. But this does bring up a good point that the benefit of having the Leica automated system is that it's a machine, so it doesn't get tired. You can run the assay overnight, come in the next morning, and cover slip, and you're good to go. When you're first implementing the ICW assay, we recommend including test slides with the primary antibody at the same concentration that was used in IHC and at a concentration that is five-fold higher. We have seen that at times it's necessary to use 2 to 5 the IHC antibody concentration for ICW. The antibody incubation time and temperature should remain the same, but I would say if you're starting this off right off the bat, just try 5 times your antibody dose because often it does require that much. Then we have a couple other recommendations for the first time you are running the Leica ICW protocol using the Leica platform. First, you should double the HIER incubation time from what it was optimal for ISH only, but at the same temperature. So typically, I think we recommend 15 minutes of target retrieval, and so doubling it would be 30 minutes. And secondly, we recommend using a protease time of 30 minutes. One final thing to say about the protease is that different kits and different tissue types require different proteases. So either protease plus, protease 3, and protease fluor. For example, BaseScope ICW requires protease 4, but BaseScope ISH only requires protease 3. So just make sure you read your manual carefully before you perform the assay. Now I'm going to go over some imaging and analysis. When analyzing RNAscope signal, it is important to know that the signal appears as numerous punctate dots of various sizes and intensities, and many clusters of dots may also be present, depending on RNA expression. It is the number of dots and/or clusters that are important rather than the size of the dot or the intensity of the dot. Each punctate dot, as represented by the yellow and pink arrows, represents one mRNA molecule. The slightly larger punctate dots shown by this pink arrow is also one RNA molecule. However, it is larger in size as more of the ZZ probes have bound to the target molecule. So what this is assuming is that this RNA transcript, although it represents one RNA transcript, was more heavily degraded, and so therefore less ZZ probes hybridized to this transcript than this RNA transcript, which is the same exact transcript, but it was slightly better preserved, so more ZZ probes hybridized to it, and therefore you have a larger dot. In contrast, this blue arrow shows a cluster. So this is kind of an oblong, slightly irregular shape, and is the result of overlapping signals from multiple RNA copies. Now, some challenges in analyzing chromogenic images. There are several things in tissue that can lead to false chromogenic signal. The images on the left are good, clean negative, and positive images. So your DAPI negative control has nice morphology, no background. Your PPI positive control has lots of punctate dots and good morphology. We recommend that you always include negative and positive controls as they provide a point of reference. The images on the right are representative images that may give a false signal. The first is an image with a pink haze that is inherent to the tissue, which may appear similar to the color in our red assays. But in a high-quality image, as seen on the far right, we are able to distinguish the colors between this pink haze and the red signal based on their intensity. This is common in tissues such as kidney that have high endogenous alkaline phosphatase activity. There are other structures in tissues that may have colors, for example here, TAR, lipofuscin, and melanin. They are easily distinguishable from red color, but they might be more difficult to separate from the brown signal if you're using DAB for IHC. After selecting the appropriate setup for your experiment, let's look at the workflow for imaging. First, we recommend using the DAPI channel to find a good focus plane. Next, go through each channel for the positive control slide and find the appropriate exposure time. This depends on your system as discussed in the previous slide, but should be less than a few hundred milliseconds with a good setup. Now you should go to the negative control slide using these exposure times to check for any background. Lower the exposure time as much as necessary to eliminate the background. We recommend acquiring images at 40x magnification or higher for analysis. For this reason, it's very important to always use these positive and negative controls to determine what is true fluorescent signal. Another tip is to only expose the sample to light while looking at it or acquiring an image similar to all immunofluorescence. Extended exposure will result in quenching of the fluorescent signal over time. And finally, we want to adjust exposure to maximize the dynamic range for each image without overexposing any pixels. Here are a few suggestions for dealing with challenging autofluorescent tissues. As I mentioned, we recommend always running positive and negative controls to provide a comparison. The green channel tends to have the most autofluorescence because it has the lowest signal-to-noise ratio, so it is helpful to assign the highest expressing target here. This way, the signal is more likely to be visible against the autofluorescent background. If you have access to a multispectral imaging system and software, this can help subtract the background by identifying broad emission spectra that are common for autofluorescence. Finally, when working with highly autofluorescent tissues, you may need to adjust the fluorophore concentrations to adjust the signal-to-noise ratio. If you have any questions and need any help, you can always contact support at support.acd@biotechne.com. When it comes to image analysis, we recommend using ImageJ, otherwise known as Fiji, QPath, or Cell Profiler. Some researchers also use Halo software to analyze RNAscope images. We have an RNAscope data analysis guide available online, as well as multiple recorded webinars that discuss analysis on these different platforms. So to add a quick summary, our ICW kit and procedures enable multi-omic profiling of tissues with single-cell resolution and spatial context. This allows us to answer complex biological questions while also preserving precious samples. These reagents are available to be automated on the Leica platform, including our RNAscope, BaseScope, and microRNAscope co-detection, and finally, multiplex fluorescent V2 assays. I want to thank you for joining us today, for joining me. Before answering a few questions, I want to highlight our previous recorded webinars on our website, on ACD's website. And I have some contact information here in case you'd like to reach out to technical support or visit our website and contact sales. We're always happy to help you with your research however we can. I want to thank you for listening, and I also want to thank the Leica team for having me, and I'm available to answer any questions you might have.
All right. Well, thank you, Blake, for your informative presentation. Now, before we start our live Q&A session, I want to draw your attention to the resource box on your screen. Here you can find additional brochures, web pages, and videos related to today's presentation. All right, so now let's get started with our live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. All right. So our first question here asks, does ER1 interfere with the ISH detection?
Okay, that's a good question. So does ER1 interfere with the ISH, so the RNAscope portion of the ICW assay? The answer is yes, it does. So ER1 is a citrate-based retrieval solution with a pH of 6, whereas ER2 is EDTA-based with a pH of 9. And we've optimized the RNAscope protocol to be compatible with ER2. So ER2 is what works best. It's what we've tested in-house and what we suggest everyone use. ER1, would it work? Perhaps, but I wouldn't suggest trying that because it's already been optimized for ER2. So yes, please use ER2 and you can try to use ER1, but I wouldn't recommend it.
Great, thank you. I have another question here in regards to ER2. This question asks, can you interchange ER2 for other retrieval types such as ER1 or ProK?
Yeah, so that's a very similar question using a proteinase K target retrieval versus ER1. Again, we recommend and we've optimized the protocol to be compatible with ER2 only. So I would highly suggest trying it with ER2. It's seeing if your antibody is also compatible, and going from there.
Great, thank you. Another question we have here. Doesn't the protease affect the primary antibody that will already be on the slide?
Yeah, so that's kind of the whole purpose of ICW. So the idea is that when you combine RNAscope with ISH, there's two ways of doing it. One is a sequential stain, where you do the ISH, and then you do the IHC, and in the ISH, there's a protease step. So the idea is that if you try a sequential stain and it doesn't work, it's likely because the protease is degrading the antigen binding site for the antibody. So the ICW is different in that we do a post-fixation step after adding the primary antibody. So this post-fixation step should hopefully preserve this epitope binding between the antibody and the antigen and protect it from the protease degradation. So the protease will still affect the antibody likely, but that's why we've developed this ICW protocol in hopes that it preserves the antigen-antibody binding more so than if we didn't have this post-fixation step.
Thank you, Blake. Got another question here that's asking, for miRNAscope and BaseScope assays, could we use another chromogen other than fast red?
So for miRNAscope and BaseScope, the fast red chromogen is the only one available for the RNAscope portion of the assay. So it would be fast red RNAscope, and then the green is for protein, because fast red is alkaline phosphatase, and that's the technology that we've developed that's compatible with RNAscope on the LICA bond. So it does need to be fast red for the ISH portion of that ICW assay.
Great. Next question asks, is the green chromogen provided by LICA?
Yeah, so there's actually two chromogens that we have used with the ICW assay. Historically, we've used the ACD green, which is sold by ACD. However, that's not fully automated, so you have to do the ICW, the ISH portion. You have to actually go offline. So for the ICW assay, if you're combining RNAscope with protein, LICA green is the green chromogen that we use. So it is provided by LICA, and it's fully automated.
Great, thank you. Next question is asking whether protein miRNA interaction can be analyzed.
Yes. Yeah, so that's kind of the purpose of this protocol. If you're trying to spatially evaluate microRNAscope expression with a protein, or to look at cell-specific expressions of microRNA expression, you can do that with the ICW assay. So yes, the interaction between those proteins and miRNA can be analyzed, and that's kind of why this has been developed.
Great, thanks, Blake. We're getting such great questions in from our audience here. We're getting so many, so we're going to keep going here. The next question that I have for you is, can the chromogenic signal from the protein detection part of the co-detection assay be used to identify fold increases in protein concentration?
Okay, yeah, so that's kind of, it depends, right? It depends on your protein and your protein concentration. If you have a protein that's already completely saturated, then if you can look at fold changes in your protein concentration without ICW, without RNAscope, then you can do it with RNAscope. So if you're using a protein stain to evaluate changes in protein expression in a tissue alone, then it's also going to be possible to do that using RNAscope and combining the two. So it depends on your protein, how large the expression is, and if you can actually quantify the difference in expression based on the protein alone.
Great. Next question. Have you compared fluorescent with chromogenic routes to see if the signal is comparable for the same transcript?
The signal should be comparable. Yeah. There is a slide somewhere. I didn't present it here today, but the protein stain is the same. So it's still the same amount of antibody binding. It's just the way that you visualize it. So yes, the fluorescent and chromogenic protein signal is comparable.
Great. Next question. What are all the possible controls should we use while trying to detect protein miRNA interactions?
So with each kit, we suggest running, every time you run RNAscope, we suggest running it with positive and negative controls to ensure that the staining is running accurately. So with the kit, there is a negative microRNA probe, which is just a scrambled probe, and then there's a positive microRNAscope probe, which is expressed by all tissues. So to answer that question, we do suggest using controls, and we have them and we sell them, and they're provided with the kit depending on which formulation of the kit you actually purchase.
All right, and we've got time for just a few more questions here. So this next one asks, are there plans to create a fluorescent multiplex kit for miRNAscope?
That's a good question. So we actually just came out with a new kit, which is called RNAscope Plus. It's not something that we have much experience combining with ICW, with protein stains yet, but I do know people are trying that. So it's not an miRNAscope fluorescent kit specifically, so it's not combining multiple miRNAscope microRNA targets, but it's actually combining one microRNA target along with up to 3 RNA targets. So you can target one very small genetic RNA transcript, and then you can target up to 3 larger, so 300 to 1,000 nucleotide RNA length transcripts simultaneously using fluorescent. So in a way, we have just released multiplex fluorescent for small RNAs combined with RNAs.
Great, thank you. This next question asks, I am interested in how the BondRx reduces a lab's hands-on time. Can you expand on this from your experience? And does this use of automation help with variability in downstream results?
Yeah, definitely. So if you have experience with RNAscope, you know that if you're doing RNAscope multiplex fluorescent or even RNAscope chromogenic manually, it's advised to split the assay up over 2 days because the whole thing is going to be around 10 to 12 hours. So if you're doing it manually, it usually takes 2 days to complete the assay. Whereas when you have a Leica bond, you can program the machine at night, press play, and then you can just run it overnight. You don't even have to think about it. So once you have everything set up on the machine, which is my job, so the field application scientist would come in, help set up the assays, and then you just run them overnight. You don't think about it, and when you get there the next day, it's done. So on the machine, it also takes around 10 hours, but that's not hands-on time. That's hands-off time. So it does really reduce the amount of time it takes to run this assay. And the second one was about variability. Yeah, so of course, if you're automating something and you're not inducing human error, it definitely improves, it reduces the amount of variability that you see compared to manual, because you know it's always going to be the same every time. As long as there's nothing wrong with the machine, your variability should be much lower.
Wonderful. Well, Blake, we're going to go ahead and wrap up with one more question here. So this question asks, I don't know if I understood this correctly, but is it possible to measure protein, IHC plus RNA in the same sample or slide for co-detection?
Yes, absolutely. That's the purpose of this protocol, integrated co-detection workflow. So you're evaluating you're doing a protein stain and combining that with an RNAscope stain. So you're looking at both protein and RNA expression on the same slide, same tissue, even within the same cell. So that's what this technology is for, and that's why it was developed.
Right. Well, as I mentioned, we're going to go ahead and wrap things up here. So, Blake, I want to thank you again for your time today and your important research. We would also like to thank LabRoots and our sponsor, Leica Biosystems, for underwriting today's educational webcast. Now, before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by the speaker via the contact information you had provided at the time of registration. And this webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, take care, everyone. Goodbye.